Well, thank you for having me. My name is Gabriel, and it is my absolute pleasure to share with you and most likely introduce most of you to the work we're doing at Enspectra Health to create an open a living window into the health and longevity of our skin. Now, I think I'm last, so I'll try to be efficient with your time. There's some formatting issues. We are living in, they say, an AI revolution in a time of massive digital upheaval. And yet when it comes to understanding what happens under the surface of the skin, we still basically have to cut it out with a biopsy. And now that's justified for disease like skin cancer, but it's generally too impractical and invasive for applications in aesthetics and skin health. What that means is that we are still largely blind to the biology that's happening beneath the surface. And because of that, we really need a better way to get at the data and the information that can personalize our skincare and our skin health journey, because you can't really personalize things if you don't have personalized data. So imagine if we could do that. Imagine if we could know exactly what's happening under the surface of the skin, non-invasively and in real time. And that's exactly the solution that we've created at Enspectra Health. This technology is so novel and new. It's actually the first new imaging physics approved by the FDA in over 28 years. And it reveals real-time histology without cutting into the skin, illuminating cellular, molecular, and even metabolic insights inside the living tissue. You can go back and forth without having to extract and kill the living cells in the skin. People are starting to take notice. There are individuals in this audience that have been imaged with our device. There are companies at this conference that are actively using our technology to deepen their understanding of skin science and to connect their therapies to living biomarkers under the surface of the skin. Maybe one of the most fun recognitions we got recently was winning an innovation award at CES with our partners at L'Oreal. Why now? Just look at this conference. We need a way to visualize regeneration. We need a way to quantify what does longevity mean. And in the same way that wearable devices and liquid biopsies have revolutionized health and wellness and diagnostics, virtual biopsy with Enspectra technology will fundamentally transform skincare and skin health, and finally allow us to translate ourselves and move from an invasive past into a non-invasive future. I'm gonna play a little video for you just to give you a sense of how the device works. It's a lot more dynamic, I think, than people realize. Ultra-portable device using a technology called multiphoton microscopy, where we scan a low-power laser through the skin. That laser excites natural autofluorescence from the molecules in the living tissue in real time, rendering a color image of the cellular structure under the surface. And those colors are revealing the concentrations of the various molecules that we all care about. It's melanin, it's elastin, it's collagen, it's and also the structure of like the epidermal thickness and the DEJ undulation. Very easy to operate. You don't need a PhD. This can be run by the medical assistant or physician assistant, and a very healthy portfolio. At this point, 20 issued patents and more pending. We've validated this through 3 clinical trials and published in some major journals. This is our pivotal trial that we used to get the FDA clearance in JAMA dermatology, basically going head-to-head against conventional histology in patients that were getting routine biopsy for suspected non-melanoma skin cancer. So on the left, non-invasive, molecularly colored, real-time Enspectra data, and then on the right, the invasive conventional histology that comes about a week later. And what we found was an over 96% accuracy and correspondence with the conventional histology, so it's truly on par with what you would get if you were to slice this off and mount it onto glass. We've also published the core technology in Nature Scientific Reports, so this is where we introduced new methodologies for how to ultra-portabilize and miniaturize multiphoton, how to do reflectance confocal and multiphoton in a way that you can image directly in the cross-section, and then validate that we can characterize key biomarkers of photo damage and age in living tissue and skin. Now, across all this innovation that we're seeing in dermatology, like, where does this fit in the landscape? So if we want to approach skin as an organ, then we need readouts across all the biological scales. And I think the classic that we know about is like the Canfield. That's where we can see complexion at the organ level from the outside. We're seeing a lot of interesting activity in the microbiome and the genomic space, but we just have this blind spot at the cellular tissue scale, and that's exactly where Enspectra fits in. So I don't think we have a silver bullet technology that solves everything, but we're the missing link that kind of connects the dots so that if you can reprogram your skin cells, make them more metabolically active, can you see new cells laid down? Can you see new structures, new remodeling happening? And then hopefully that should be reflected in the mirror or the camera. So where do we go next? So we are a startup. We are looking for partners. We are looking for investors. So if that is you, I hope you brought your checkbooks. But seriously, we believe that the groups that align with us will basically corner the market on in vivo biomarkers because we're that differentiated. And one of the things that we're working on for the next phase of our development is metabolic sensing. So when you look at these beautiful pictures of our device, those colors come from the wavelengths emitted by the different molecules. You can also look at the timing parameter of those photons. And if you look at the timing parameters, it tells you the binding state of the metabolic molecules in the cytoplasm, particularly NADH and FAD. And so you can get direct metabolic readouts in living skin that would tell us if we're actually boosting cellular energy, are we boosting mitochondrial activity, and then through our existing type of imaging, does that result in beneficial remodeling? We have to be able to digest this. We get a lot of data. One of the advantages of having a one-of-a-kind device is we have one-of-a-kind data sets, and then we make AI models to simplify that interpretation. We have two basic varieties, biometrics, which is very specific. It basically takes those images and automates key markers like how much elastin and collagen do you have in the dermis, DEJ undulation, epidermal thickness, melanin concentration in the epidermis and dermis. And then Dermatype takes all of that and tries to generalize. So it makes a fingerprint, basically what we call a vector embedding of all of those metrics of your skin. And then the idea is that as we continuously scan more and more people, we'll basically build a search engine for skin to literally find your skin twin. Who are people like you, and how do they benefit from similar therapies? Finally, all packaged into a format that you can deliver to patients to truly personalize their care, helping us finally move from guesswork to precision medicine and skincare. So I will be here during the networking break, and I would love to talk with you if you'd like to have any more information. Thank you.